Bismillahirrahmanirrahim. This is a video lecture on the introduction to real-time PCR or quantitative PCR, QPCR. Nucleic acid amplification and detection techniques are among the most valuable tools in biological research today. Scientists in all areas of life science, for example, in basic research, biotechnology, medicine, forensics, diagnostics, and many more, utilize these methods in a wide range of applications. Routine diagnostic lab also use these methods, especially the real-time instrument, real-time techniques to amplify, to quantitate certain types of viruses, for example, human hepatitis virus, HIV, cytomegalovirus, and some other medical importance virus. So, standard PCR usually can give qualitative methods. That means the result is either positive or negative. So that means it detects the presence or absence of certain type of pathogens. But real-time PCR can be used for both qualitative and quantitative analysis. This is the instrument of standard PCR, the thermocycler. And this one is the machine for real-time PCR. We usually attach to the computer or PC. Let's look at what is the meaning of PCR. Polymerase chain reaction or PCR is a biochemical process for amplifying specific DNA sequences from relatively small amounts of starting materials. So, for example, in a samples, we have a very small target DNA. After using PCR, it doubles the amount or multiply the amounts of the original target DNA target to many folds of the DNA target. So we can easily detect and we can easily analyze the target gene. And inside the PCR, the process, we must have reagent to make the DNA. These are the PCR components. So what are the PCR components? So first we have the primer. The primer is a short sequence of DNA that can specify which target that we want to amplify. And then we must have nucleotides or the deoxynucleotides. The adenine, guanine, cytosine, thymine. These are the building blocks of the DNA. Without them, our new DNA cannot be made. And we must have the most important enzyme in PCR, which is the Taq polymerase. This is an enzyme that involved in elongation process. It can build the DNA by the presence of these nucleotides. And then finally, DNA. This is derived from our samples. So in PCR cycles, we have 4 steps. Number 1, initial denaturation. Secondly, denaturation. Thirdly, annealing. And lastly, extension step. So usually, denaturation, annealing, and extension, these 3 steps will be repeated until up to 30 to 4 cycles to make a very large amount of target DNA. So what happened during these steps? Let's look at the diagram here. This is our samples. The extracted samples. Our samples, for example, serum, urine, tissues, and so on, must be we must extract the nucleic acid first before we run for PCR. So depend on what type of the nucleic acid, either DNA or RNA. If our target nucleic acid is DNA, we must use DNA extraction kit. But if we want to, let's say, amplify RNA virus, we must use RNA extraction kit. So let's look at the step. The first one, denaturation. What happened? We increase the temperature usually up to 95 degrees Celsius. So the temperature is increased to separate DNA strands. So in this step, the double-stranded DNA unwind into single-stranded DNA. The next step is the annealing step. Okay, we lower down the temperature, as usually 48 to 72 degrees Celsius. So temperature is decreased to allow primers, our short sequence of DNA that determine our target DNA, to base pair to attach complementary to the DNA template. Then we increase some more temperature, usually 68 to 72 degrees Celsius for the extension process. So in this step, the polymerase bind to the DNA strand, and with the presence of deoxynucleotides, our adenine, guanine, cytosine, thymine, the building blocks of the DNA, will bind and attach and form the new DNA strand. Okay, this is what happened in one cycle, and this cycle will be repeated up to 30 to 40 cycle. So we will get abundant of target DNA. So we can easily detect, we can easily analyze. Okay, we send for sequencing and so on. So depend on our objective. Okay, for standard PCR, analysis is done based on the endpoint analysis. That means once PCR process is completed, we must further with the gel electrophoresis. Okay, gel electrophoresis, we run the gel, and we observe under UV light by using an instrument called gel documentation system, or GelDoc. Okay, this is an example of research: antibiotic resistant genes and susceptibility patterns in staphylococci. So the researcher used several genes to be amplified by using specific pair of primers, and then we already know that we already determine the size of amplified product. Okay, this is what happened in the standard PCR. So once PCR process is completed, the amplicon, the product of PCR cycles, is called amplicon. So the amplicon will be run through gel electrophoresis, okay, to separate the band, the size according to the molecular weight, okay, the base pair. Sorry, not molecular weight, according to the base pair, because we can separate the base pair according to the electric current. So usually on the agarose gel, okay, usually we use agarose gel. If we do the protein, we must use polyacrylamide gel, okay, different type of gel. So our nucleic acid, the DNA, we can use agarose gel to separate the DNA. So in this image, this agarose gel consists of several lane. Okay, so this is well, and this has 15 lane. That means about up to 15 samples can be used. Okay, in lane 7, okay, this is the DNA marker, 100 base pair DNA marker. This the DNA molecular size marker, 100 base pair. Okay, that means every band here is 100 base pair. Okay, it start with this one. 100 base pair, 200, 300, 400, 500, and up to 1000 base pair. And then we run our gel, agarose gel, and if the band is produced, we must compare with the band, the ladder here. So for example, this gene APH3 will have the target DNA band 242 base pair. So we observe according to the DNA marker. So we know that this is the band belongs to this gene. Okay. And then let's look at this one, this band. So it is situated in between 400 to 500 base pair. So actually this belongs to this gene, AAC-APH2. So based on the gel electrophoresis, we know our product, okay, derived from which gene. But in real-time PCR, no need to run gel electrophoresis because our analysis can be done in the software. By using a software, we can look for the amplification curve and the standard curve and the melting curve analysis as well. Okay. Actually, from the standard PCR, we can see the scientist has come with different variants of PCR. Okay? From the standard PCR, we can have the real-time PCR, multiplex PCR, nested PCR, and many more. Okay, all this PCR. Okay, for example, reverse transcriptase PCR, transcriptase. Okay, for RNA virus, for example. Okay, all these variants of PCR use the same principle and same mechanism as in the standard PCR. Okay, for example, in real-time PCR, okay, we just add the fluorescence, the dye that can be analyzed at real time during the PCR process itself. Okay, now let's look at what is real-time PCR. Okay, real-time or quantitative PCR, QPCR, is a standard PCR with the advantage of detecting the amount of DNA formed after each cycle with either fluorescence dye or fluorescence-tagged oligonucleotide probes. Okay, fluorescence dye means the cyber green. Okay, fluorescence-tagged probe means the Tagman probes. So the qPCR results can be obtained faster and with less variability than the standard PCR due to the sensitive fluorescence chemistry and elimination of post-PCR detection procedures. That means no need to run gel electrophoresis because running gel electrophoresis will take approximately about 1 hour. Okay. And also, real-time PCR is more sensitive. Okay. Real-time PCR is more sensitive than the standard PCR. So usually, real-time PCR is useful for the investigation of gene expression, okay, viral load, the quantitation of the DNA inside the samples, and then pathogen detection and numerous other applications. Okay, measuring fluorescence. As we know, the real-time PCR use the fluorescence dye. So the fluorescence need to be measured and detected by the instrument, and then the instrument will plot the amplification curve. So we can analyze the results based on the amplification curve. So measuring fluorescence, the intensity of the fluorescence emitted during qPCR correlates to the amount of DNA product formed. So more DNA product formed, more intense the fluorescence signal detected by the machine. Okay, fluorescence exponentially increases as the DNA template is amplified. After a few cycles of qPCR, fluorescence surpasses a threshold level set above background fluorescence and starts to increase exponentially. Okay, this is a threshold level or baseline. Okay, threshold level separates the fluorescent signals from the fluorescence derived from amplification and the fluorescence derived from the reagent. Okay, so we must separate them in order to in order to get specific results. So fluorescence signal exponentially increases in qPCR. Okay, threshold levels, the point at which fluorescence emitted from PCR product formation is significant from background fluorescence. So exponential phase, okay, this is the exponential phase. So the fluorescence increases exponentially as the DNA target sequence is amplified. Okay, this is the cycle, up to 50 cycles. So more cycles, so more fluorescence, more DNA will be produced and more fluorescence signal will be emitted until it reach the plateau phase. Okay, this is the plateau phase. So in plateau phase, the fluorescence signals levels off and any changes in DNA concentration can no longer be recognized. So actually the analysis is based on the exponential phase. Okay, real-time PCR and CT values. CT means cycle threshold. Okay, in real-time PCR assay, a positive reaction is detected by the accumulation of a fluorescence signal. So the CT or cycle threshold is defined as the number of cycles required for the fluorescent signal to cross the threshold. For example, exceed the background level. Okay, this is a background. It is called background. Okay, it is actually our reagent that contains the cyber grain, for example, and so on. So the fluorescence dye. So we must put a line to distinguish between the fluorescence derived from the reagent and the fluorescence derived from the amplification. So the CT levels are inversely proportional to the amount of target nucleic acid in the sample. For example, in the lower CT level, the greater the amount of target nucleic acid in the sample. Okay, for example here, okay, so this is the CT value, okay, where the amplification curve surpass or cross the threshold. Okay, the green color here is the threshold line. So two samples here, one CT value is 21 and another is CT 39. Strong positive. Okay, this CT value 21 indicates strong positive because it requires only about 21 cycles to produce signals, but this samples shows weak positive because it requires more cycles in order to give signals, okay, to the instrument. So it requires up to 39 or to 40 cycles. So it means it is a weak positive. So this one is a strong positive. So let's look at this table here. CTs or CT value less than 29, a strong positive reaction indicative of abundant target nucleic acid in the sample, okay, before the amplification process starts. Any CT value 30 to 37 are positive reaction indicative of moderate amounts of target nucleic acid in the sample. CT value of 38 to 40 are weak reaction indicative of minimal amounts of target nucleic acid, which could represent an infection state or environmental contamination. Okay, usually, weak reaction need to be repeated until so we can confirm it is a real weak reaction and no contamination from the environment. Okay, fluorescence detection chemistries. Okay, generally there are two types of detection chemistries utilized in QPCR, in real-time PCR. The first one, cyber-green-based chemistry, and another one is the probe-based chemistry. Okay, cyber-green-based chemistries use fluorophores or fluorescence dye that can intercalate and bind with any double-stranded DNA present in the sample, in the reaction. So the fluorescence signals of the intercalated dye is several orders of magnitude higher than that of unbound dye. So that means the free cyber-green dye, when floating freely in the reaction mixture, has low magnitude of fluorescence, but if the dye is attached or intercalate in between the DNA strand, so the fluorescence signals is stronger. So the most common fluorophore used is cyber-green dye. So secondly, the probe-based chemistry utilizes fluorescently tagged oligonucleotide probes that are specifically designed to detect and target the DNA sequence. So these probes rely on the principle of fluorescence resonance energy transfer, or FRET, to generate an increase in fluorescence. So the most common target-specific probe used is a hydrolysis probe, the TACMAN. So in between cyber-green and the hydrolysis probe, so the hydrolysis probe will give more sensitivity, okay, as compared to the cyber-green. So let's look at the diagram here showing how the TACMAN and cyber-green works. Okay, in TACMAN probe, we have similar, the primer, the forward primer, and reverse primer. And we have another short sequence of the DNA, which is tagged with two types of dye. The first one is the fluorophore, okay, the reporter. This is called reporter. And another one is the quencher. So if these two dye is attached together, the fluorescence signal is very low because the reporter has a fluorescence energy, but the quencher will absorb the energy. So the reporter need to be released in order to give signal so that the PCR instrument can detect. So what happened here? During annealing step, so the primers, the forward primer bind, the reverse primer bind to the single-stranded DNA, as well as the probe. Okay, the DNA sequence with the probe. So during the extension process, DNA polymerase will elongate the DNA strand until it reaches the probe. So the DNA polymerase has a mechanism which is called exonuclease activity. So it is some sort of they can eat DNA. So the DNA polymerase will go through the probe and finally release the reporter from the quencher. So once the reporter is released and excites, so the fluorescence, the reporter can emit the fluorescence energy, so that it can be detected by the instrument. So more reporter is released, so the signals is stronger than a small amount of reporter release. Okay, cybergreen. In the cybergreen, freely floating cybergreen has a low energy. Okay, after several cycles, new DNA strands, double-stranded strands produced. So these cybergreens will bind to the double-stranded DNA, and the signals, the fluorescence signal, the fluorescence signals is amplified. More fluorescence signals is sent to the detector, so it can make the graph increase curve. So before attached to the double-stranded DNA, the signals is very low, but once the cybergreens attach or bind to the double-stranded DNA, so the signals is signifies. So the real-time instrument is actually measure the emission filter, the emission light from the fluorescence, and then it requires the specific wavelength. Okay, and then the fluorescence is emitted and measured by the light detector inside the instrument. Okay, fluorescence detection in real-time PCR machine. A light source produces light that is filtered for specific wavelength required for excitation of fluorophores in the reaction. And then accumulation of PCR products is monitored by observing emitted fluorescence. Okay, in this step. Okay, reflected excitation light is filtered before detection, and only fluorophore specific wavelengths are allowed to pass the detector. So from the detection, the amplification is plotted. So if we use cybergreen detection method, we must add a step which is called melting curve. Okay, melting curve analysis. Okay, melting curve analysis is done to check the specificity and the validity of our PCR process. Because the cybergreen dye can bind to any double-stranded DNA. So by using melting curve analysis, we can specify whether the amplified DNA is derived from our target DNA or from other sources, for example, primer dimers or any environmental contamination. So let's look at the video for the melting curve analysis.
Are you using cybergreen chemistry for your real-time experiments and getting multiple peaks in your melt curve? And are you asking yourself, is this a bad thing? Let's start with a little background. Unlike yours truly, CyberGreen1 chemistry is a free-floating dye. Here's how it works. When cybergreen dye is just swimming around in the tube, it doesn't give off much fluorescence, even when we zap it with a light source on a real-time instrument. But cybergreen dye really likes to bind to double-stranded DNA, and when it does, and then you hit it with light, the dye gets excited and fluoresces. At least in theory then, the basic idea is this. As PCR creates more and more product, the signal of cybergreen dye goes up proportionally. In practice though, this doesn't always happen. That's because cybergreen dye binds to any double-stranded DNA, meaning every double-stranded molecule in the tube will bind cybergreen dye and add to the fluorescent signal. Because of this concern, users run melt curves after each experiment. They do this by slowly raising the block temperature from about 60 up to 95 degrees Celsius and monitoring fluorescence. As you can see, signal drops slowly until at some point it drops off suddenly to 0. halfway down this drop-off is the presumed melting temperature of the product created during PCR. If you choose the derivative view from this drop-down menu, the drop-off gets converted into a peak. What we're hoping to see is one clearly defined peak which suggests, doesn't prove, mind you, but suggests that you have a clean amplification of a single product. One thing you most definitely don't want to see is multiple peaks, as this suggests your amplification curves are a composite of more than one product. Not a good thing. So what causes extraneous peaks? Well, really depends. Often they're caused either by non-specific amplification or primer-dimer formation. In the first case, you'll need to redesign your primers to a more specific sequence. In the latter case, you may simply need to lower the concentration of primer to discourage dimer formation, although a primer redesign may ultimately be necessary. But multiple discrete peaks are not the only thing that should concern you. Other anomalies such as asymmetrical peaks, shoulders, and unusually chunky peaks should make the user question the validity of his or her data. The problem is, it's difficult to know exactly what's causing certain anomalies, so some users end up spending a lot of time repeating failed experiments under multiple new conditions or with multiple new primer sets. That's neither fun nor affordable. Still, cybergreen chemistry is perfectly valid for qPCR when all things go well. Users just need to take extra care in designing their experiments and take additional quality control steps when evaluating their data. And remember, you can submit your questions on Facebook, Twitter, or on lifetechnologies.com/AskTechman.